our group is going to show some progress that he made on the linksproject.org website. Ned, are you on?
Hi, everybody. This is Ned. Yes, I am on.
So if you can share your screen and start, your just give an update on some of the changes that you're working on.
Yeah, sure. Let's see. Can everybody see my screen?
Yes.
This is on the Links project. Okay, so I'll get started. Hi, everybody. I'm Ned, and I'm the developer for linksproject.org. Currently, what we have on the development site for linksproject.org, what is different from production is that we've added this section right here, which features upcoming events. And so essentially this is just a banner. We've added scrolling so that way we can show some new events. So currently we see here that we have an upcoming AACR event on April 4th, as well as a data symposium in May. So also the second update, as we scroll down from the home screen, are some announcements. These announcements are now database driven. So whenever we would like to add in a new announcement, and this is anticipation for the Links Project mobile app as well, we can directly add it into the database, and we can see here a few announcements. Other progress, we've made changes to the community page. So if we were to click here, now we have many featured events broken down by categories. So some of these include crowdsourcing challenges, conferences, courses, symposia, training programs, and webinars. So by default, all of these community events will be showing all in the most recent order, in the order that is the most recent up to the latest. And if we were to click on crowdsourcing challenges, it will filter these events based on the upcoming events. Right here you can see, as well as the past events that we've had. And this goes for all the other tabs as well. And so we also have featured events. So this AACR event is featured, and this is what we have at the moment for this one. And the last item, just a few tools that we've added in for the Links transcriptomics. We've had 5 new tools. So some of these include Morpheus app, ICV app, repurposing, as well as the touchstone, and we've also changed the tool from Links Cloud app to the Clue platform, which all links to clue.io. That summarizes most of the important changes that we've had to Links project. And so if everybody likes these changes and agrees, we can push it out into production. So you can check out all these changes on the development site at amp.farm.mssm.edu/links, which is the link right here. And that's all I have to present.
Great, thanks.
This is Max from the Broad Institute. Thanks a lot for making those changes so fast. We're really excited that Clue is now up here, and
great.
We've yeah.
Oh,
it looks
thank you.
great.
Hi. This is Terry from Neuralink.
Yes. Hi,
so
Terry.
Great changes. Thank you. I have a couple bugs that I found, and then one suggestion basically on this tools page.
Mm-hmm.
And so since you're on the tools page now, I'll just hit that one first.
Okay.
So you guys, we're producing, there's so many tools. I mean, it's awesome how many tools we're starting to get here. Mm-hmm. I'm wondering, I see that you have a filter, but
yes.
I'm wondering, I guess, and you could do it by role, but I was just making sure that there is a way for a user to come here, because it's kind of overwhelming when you just see all these tools and you have no idea what each one of them does. Mm-hmm. There's a way to categorize them, and maybe that's all under role.
Yes.
Way to do it.
Right. So role are the categories for the tools that we asked different centers to provide. So this is all information provided by the creators of the tools. And so most of these tools, if we were to hover over this icon over here, such as this eye, or some of the, every single tool has some categories. And so we can identify which tool comes from, has what data type, what role, and what features. And so any
Okay.
user who is interested in their specific, they know what they're looking for, they can hover over these data type, this icon over here, and discover what the tool can be used for, as well as you can use this graphic interface here to filter different tools.
Okay, perfect. Thank you. That's awesome.
Thank you.
So my next comment actually happens to be about it's about a linkage and not showing some data when you go from page to page. So if you go back to your home page.
Sure.
And then you click on Discover Links Data. I just want to see if it does it for you, because this is what happens for me. Discover
Okay.
Links Data.
Mm-hmm.
And then if you just go into the second column and just scroll down. Don't click on anything. Just scroll it.
Mm-hmm.
If you do you see ATAC Seek anywhere in that list? Because I don't see it.
You're right. I don't see it here.
Okay, but here's the deal. So if you click on, because that's under methods, that's highlighting the list under methods, right? And if you click on subject areas or centers and then go back to methods, ATAC Seek comes up.
Mm-hmm.
So
Okay, and then go back to methods.
It
Yes,
should show
okay,
up
I
there.
see it here. Okay.
So I hope that's happening for other things as well. But I
Okay.
did notice that for
Okay. Well, sure. We will look into this. Well, this is actually taken care of by the presenters, perhaps upcoming presenters later on today. But yes, we will make note of this. Thank you so much.
Ned, just a quick question. This all looked great to me. For the conference information that's provided there, as Detox, we're planning a few workshops and presentations in the upcoming meetings, talks meetings.
Okay.
Do we still just email you or Sherry?
Yeah, you can just hi everyone, it's Sherry. Yeah, you can just email me any of your upcoming events, activities,
Mm-hmm.
Cool. All right, thanks.
and I'll take care of it.
Any activities, you just send them over. We'll take care of it. Okay.
Okay, thanks, Ned. I guess we will wait until maybe the middle of next week. Yeah, I don't have like a specific date to give, but if you send us any suggestions for corrections, if you can review this material, so we don't want to put it up before review, but once everyone approve or if we don't hear from you, we will just push it by middle of next week onto the live site. So the next presentation today is by Caroline Montiero. And I remember that if you remember last time that we met, we said we're going to share a table with the canned analyses. And then what happened, we reviewed this table and we realized that it was not ready to be shared. We had internal things that we had to fix. So Caroline is going to talk about her work of creating and setting up that table. She'll give an update of where that part of the project is and a little bit of a refresher of this idea of this canned analysis.
Can you see my screen?
I cannot see your screen. Okay.
Hi, can everyone hear me?
Yes.
Good afternoon, everyone. Let me just start this. My name is Caroline Monteiro. I'm here with the MyLab, and today I'll speak a bit more about the canned analysis, but now we'll approach it from the metadata side. So first, a quick recap. Canned analysis is a pre-packaged analysis of a specific data set by a specific computational tool, which is stored online and can be accessed on a web page. and it's characterized by the dataset URL, the tool URL, and the can analysis URL. What we'll do now is add this additional layer of information to provide context and describe the content of this object. To do this, the information was organized as spreadsheets, one per center, that could be further adapted for data and center-specific traits as needed. But instead of going straight to the spreadsheet and show you that, I'll start by going over its fields and its structure so we can all really understand what we chose to, metadata we chose to collect. It was important that this descriptive metadata layer captured the key elements of the data, such as the relationship between dataset and tool, center, and the original source of the data and metadata. So this diagram shows an overview of all the fields we'll be collecting and are collecting to identify, describe, and locate each of the scanned analysis objects. And for the creation of this particular set of sheets, the metadata was acquired manually, but in the future, with the exception of the elements here shown in yellow, the metadata will be acquired automatically from the data. And this will enable us to maintain a consensus across the can analysis objects, regardless of tool and data type. Next, I'm going to show you all the source used to populate each of these fields. Our search was center-specific, so each line in the spreadsheet shows a tool the center used to analyze its own datasets. I'm using an HMS dataset and tool here as an example. So the first three columns, center dataset ID, and tool, came from the center's own website. So we have HMS, the ID is 2120, which is the HMS dataset ID, and the title and the tool is this drug pathway browser. The links IDs, both LDS IDs and LDG IDs, come from this links dataset registration spreadsheet that is just shown here in pink. And the tool icon is what we're using now on linksproject.org, and of course, this can be easily changed. So the title is the title defined by the center for this dataset. One problem we had previously was the centers and with different titles, so we are also going to keep the original title for each dataset. The canned analysis URL here is the direct link to the analysis results or the analysis page. For the three fields under publication, if the tool, the dataset, or the can analysis has been published, we'll provide the IDs or links to it here. And you can see in this example, this dataset was mentioned in these two publications, so we have the PubMed IDs here showing. We also collect information on the center's principal investigator and any other investigator responsible for this data. For these two fields here marked in yellow, namely canned analysis description and keywords, to better annotate it, we would like to ask the centers to provide this information. Since sorry, my headphone came off. So the canned analysis description should be a brief and specific description of the analysis object, and is specific for that tool and dataset. The only constraint we have is this maximum character length of 200 characters. The keywords will be used to enrich the search and should be separated by a semicolon, and this field accepts both single words and specific phrases as keywords. Finally, after all the metadata has been collected and of course reviewed, the canned analysis will be added to linksproject.org, where users can already see all the tools that we have available, but now we'll also have a straightforward view of all the feedback analysis that we have across our centers. And here is a mock-up of a canned analysis object. Not all the metadata collected is shown here, but rather it will be used to enrich the search and facilitate the discovery of objects, making it easier to find, access, interoperate, and reuse both data and tools. So overall, this is what we have right now. I'll add the link to the spreadsheet to the chat. There are some specifics for each center that I don't should really spend a lot of time going into it here, but we can talk about them if there's any specific questions. But all the definitions are added here to the top and should be pretty straightforward to understand. And that's pretty much it. Are there any questions?
This is Liz from HMS. So you said that this document so far has been populated manually, just
Yes.
by going through the websites? Okay. That example that you showed for HMS is the same one you showed last time, and that tool is not linked to that data set. So I'm a little concerned about where this information is coming from. I mean, there's nowhere that I'm aware of on our site where that linkage is made.
Well, I can actually.
I mean, probably best that we talk about this offline, but I think in general, definitely all the centers will need to check the information very carefully.
Yes, usually we go through the tool and there's a bar under the page saying the data sets and signatures where it came from. And that's where this data came from. But we can, of course, and that's why we're giving it all to you to, check.
Yeah, I mean, for this particular one, it refers to this paper Heiser et al. From OHSU from a few years ago, and the data set is related to that paper, but the data set and the browser are not related. So that's, anyway, we'll just have to check everything carefully.
So Caroline, what is the timeline when this table is going to be shared with the centers?
I could potentially share it now, and I would really like them to go through all the data sets, but seeing that, Liz just mentioned this specific data set, I would like to go, through them once again. So probably on Tuesday, it should be okay to share this table.
Okay. So I guess, does anyone else have any questions before we move on to the final presentation by Amar? I guess not. Amar, are you ready?
Yes.
So Amar is the developer of the links data portal that was brought up earlier, and he's going to give us a summary of the work that he's doing on that. And I may have to leave in the middle because I have to move to another call. So, Stefan, you can take over later.
Yeah, sure. Amar, why don't you grab the, yeah, okay, sounds good. Okay.
So, so I'm Amar. I work with Stefan Schurer in University of Miami. So today is to give a brief update about what we, about the links portal, the recent changes in the links portal.
Maybe best to put into presentation mode if it's not already, I'm not sure. Oh, it's Adobe anyway. You can probably do that.
Okay. And so links portal is the primary portal for all the links data, and the goal of the portal is to provide an integrated access for data, for the links data using search, browse, and other ways. And the primary audience for this portal is going to be a biologist who is interested in links data, or a scientist who wants to explore. So in order to get to the links portal, what you could do is once you go to the linksproject.org, you have an option to directly go to links portal. Once you click on that, it takes you to the links portal, links data portal. So we redesigned the portal in last few months, and based on the feedback that we collected from the community, as well as by visiting various websites. So we collected the feedback, and based on the feedback, we have improved the navigation. So once you go to the landing page, so this is now the main landing page. As soon as you go to the landing page, the links data portal, the links data portal is an unified resource, right? And it is a suite of many apps, right? It comprises of many apps. So what we did was, in order for easy navigation, we included all the icons on top of the portal. So as soon as you go there, you'd see these 5 icons, which actually point to the different apps that we have. And the next section below the icons you see is a browse section, which you could browse using various methods. And underneath that, you have a description of each essay. So for example, if there is a non-links user and he wants to know what this is about, then he can directly click on the essay tab to see the description of the essays. And we have on these icons are of different types, right? One of them is data sets, and the rest of them are different entities. The main goal of today's presentation is to show the small molecule app. So that's the one here. That's the one which has stars. So on the landing page, so these are some of the new changes in the landing page. On the landing page, what we did was we added publications whenever there is available. For example, there's a publication available for a HMS data set. We added it here. And right now, so instead of downloading the package, if you want to download the metadata of the data sets, you could directly do it from the landing page itself. So once you go, click on the metadata tab, and there's a download button on top of each particular entity type. So for example, there's a cell, you have the download on top of that. And we also added like experimental metadata to the dataset landing pages, which will give you what kind of concentrations and time points this dataset is actually tested at. And all of those, all the experimental conditions, time points, concentrations are in here. This is just a brief summary. It doesn't have all the details, but it has the different concentrations and time points. And the next, so from here we can go to the small molecule landing page. So if you're on the landing page, you can click on the metadata tab, and if you have a small molecule, you can click on the small molecule. You'd be directly taken to the small molecule page. And this, what we also did was we have changed a lot of infrastructure for the small molecule. We have changed the way we were registering and standardizing the small molecules, because we had a lot of issues in the way we were desalting the chemicals and smiles and stuff like that. So we have redone the entire standardization pipeline, and based on the standardized pipeline, we have also collected data from Campbell and KKB, like Campbell and KKB, aggregated all the metadata. We curated metadata for those small molecules. And on the landing page of the small molecules, you'll see a summary statistics about what is available, like what are the different things available, and how you could search the system using with some examples. And on the curated tab, we have added a new tab called curated, which gives you a description of all the drug summary, gives you mesh terms, EFO terms, mechanism of action for the compounds, and the genes associated with the mechanism of action, as well as the FDA phase of the compounds, and the drug classifications. All of those you'll see on the drug on the curated tab. And we also collected bioactivities based on aggregated from Campbell, and when we aggregate, we have, so there are like millions of points in the Campbell. We have aggregated them and filtered them so that only the relevant ones would be shown in the portal. So we now have around 200,000 data points, and for about 13,000 compounds that Links has. And the bioactivities are for on and off target mechanisms. So it's not just concentrated on the mechanisms of action, but even if it is not included in the mechanisms of action, we search for those, get the aggregated data and put it into the system. So that's, I'll give you a brief demo about how the system is. So this is the Links data portal, right? And you can see when you click on any of these essays, you can see the description. Once you click on the essays, you also see the changes in the counts here. So, for example, if you click on kinome scan, you'll see the change of so there are 147 small molecules which are tested in kinome scan with zero cell lines, right? So this is kind of things that you'd see. And once you click on those, it would directly take you to the small molecules which has tested with kinome scan, which are tested in kinome scan data set, kinome scan assay, which are tested with. So let me go back and click on the small molecule. This would give you a summary of what we have. So this is the brief summary of about the small molecule page, and there are some examples here. So this is the search. There's a search bar, there's a browse, and there's about, help, and substructure search. And underneath the search, you have a section describing what you could search in that thing. There are a few examples here, and you could also explore the search. You could also use the search to explore using the annotation. So you could say, okay, I only want kinase inhibitors. So I only want cyclin-dependent kinase inhibitors. You could do that. So I put an example here. I'm just going, and we have a statistics here showing by the source, by the center, and also by the different drug phases how the compounds are in. So when you click on I'm going to show you one example, cyclin-dependent kinase inhibitor. You could use mesh terms or even mesh IDs or EFO terms or EFIDs or like the mechanisms, the particular mechanism actions to search. So here we have two things. We have two views. There's a list view and table view. On the one side, you see the structure of the image, structure of the compound. On the other side, you see the number of data sets the compound participated based on the subject area. So based on the study. So for example, this salicylic is tested with in 18. So you should see it here. It's tested in 26 data sets, and of the 26 data sets, 6 of them are transcriptomics data sets, and 17 of them are imaging data sets. And we have few links to go to PubChem, Campbell, and KB. And once you go to the tab table view, it's the same data, but you could see the 22 cell lines. You could scroll over to see all the cell lines. You can click on one of them. It will take you to the exact cell line, the MCF7 cell line that you selected here. And you could even go to a data set using that, right? And so once you go back to, once you are on this page, go to this landing, click on the icon or on the name, it will take you to the landing page of the small molecules. And the landing page has 3 tabs. It has description tab, which has the basic descriptions. So in the descriptions, we have added batch information, which is what the centers actually provide us, right? So these are all the information that the centers provide us, which we were not able to do before, but now because of the change in infrastructure, we were able to do that. And you'll see which, so if it is an LSM ID 1001, these are corresponding IDs from various centers, which were standardized to become this LSM ID. And you could click on any of those. It would take you to the right field. And then you have the data sets. So this salicylate is tested in about 25 data sets. You could see all of them here. In the curated tab, we have a summary based on the drug. We have a drug summary, mechanism of action summary, and bioactivity summary. You could sort it by any of these values, or you could search by, for example, you could say C and A. You could search and filter by them, right? And one, it would filter those that have those values. You could further filter them, and you could say, for example, if you have these ones and you want to get more description about it, you can click on those. It will take you to the ontology where you have the EFO term described in more detail. So, and on top of, so as you see, on top of the summary on the page, what we have added is all these icons that you saw on the landing page. So these are the icons that you see on the landing page are now part of the small molecule page. So by clicking any of those, you could come back, you could go visit to that specific app that you're looking for. So looking for small molecules, you go there, right? So, and right, so this is about the demo. You have any questions, please ask me.
I had a couple questions. This is Liz from HMS. First, about the dataset landing page where you have the concentrations and time points. So are those the concentrations and time points that the centers checked? And so is that all updated for that?
So right now it's on dev. So we'll ask centers once it's ready to move to production. So I think we haven't sent any emails to check that.
So Liz, I think, so this is Stefan, Liz. I think those are those that we curated, but then we put them in this big Excel document in Google, and we asked everybody, and probably for the last 3 data working group meetings, to basically review them and make sure it's correct.
Right, that was what I was referring to. So it's the curated version that's on the dev site right now?
Yeah. So this is not yet public, right? So I think what we could do is, it may make sense to give everybody the opportunity to take a look at the content again, if you want to, before we put it public, before we put it into production. Okay. But those are the ones that we basically curated and then you reviewed. And I know for sure you reviewed them. I'm not sure if everybody else reviewed them. And then the other thing is then, of course, as you all know, as the experimental metadata are going to be collected in a more systematic way, then this will of course be replaced by that information, right? Right. And then it will be a formal process that Amar will also show.
Okay, so I'll just check a couple of those, but it's as long as it's supposed to be the updated versions, then it should be fine.
Yeah, just make sure about it is actually, but it should be, you know.
And then I had another question, so about the small molecule pages, the detail pages, basically. So I'm a little concerned about calling the target information mechanism of action. I mean, you know, a big thing in links is we understand the polypharmacology of these drugs. So, you know, mechanism of action is kind of a...
So the mechanism of action targets, they're actually, those are the annotated mechanisms of action of drugs. Those are annotated in, you know, what's basically FDA, and, you know, this information is collected like in some resources, but it's basically the FDA understood mechanism of actions of a drug. If that's not known or not there, it wouldn't be in this mechanism of action column. And then independent from that, I think as Amar mentioned, all the known targets are there. Now, I understand that, you know, as our understanding of drugs may evolve, right, that maybe this could be, you know, there could be more knowledge, right, about what really, if there's a polypharmacology action or something like that, right? But basically, those are the ones that are kind of the officially annotated FDA mechanisms of action.
So,
Yeah, so
where exactly is the information coming from?
So this is coming from FDA. So,
And is mechanism of action the term that they're using?
Uh-huh.
That's a concern for FDA then. That's a problem.
Right, for how the effect, how the you know, pharmacological effect of a drug is mediated, right? Kind of the mechanism of action target.
Right, but I mean, we know full well that some of these drugs, what could be called the mechanism of action or what it was designed against in the first place is not how it's actually having efficacy.
So again, right, this is basically what's coming from the FDA. If there's new information, right, that's very current, that's not reflected in the mechanism of action that's described at the FDA of an approved drug, that it will not be here.
My concern is not with the information, it's with the term that's being used. Now, if it's coming from FDA, then that's an FDA problem. I'm not sure we want to propagate that problem, but I mean, if it's explained somewhere that term is being used here in links because that's the term that FDA is using, you know, I think that's okay.
I mean, we could specify it more, but basically at the openfda.gov, you know, you can see it's basically the farm class MOA. It's called mechanism of action. It's the definition: molecular, subcellular, or cellular level function activity of a drug product pharmacological class. Anyway, so I mean, we could specify it and say specifically FDA mechanism of action. Maybe that would be better, you know. So then it's not being confused with the rich biological research that's perhaps coming out of links, right? And that we may not want to confuse. So we could update that, you know, that make it more explicit.
So obviously, I haven't played around with this because this is just on the dev site. But I mean, I think this sort of touches on a larger issue, which is if you are pulling information in from somewhere, or you're pulling, you know, terms and those sorts of things, I think it's very helpful for there to be somewhere clearly accessible on the site, like a README that says, you know, this is where the information came from. This is, if you're not directly linking out. I mean, I saw you directly link out to Kemble in some places and things like that, but you know, somewhere where people can go to know unambiguously where the information and the terms came from.
Yeah, that makes sense. We should have, it should be clear where it's coming from. That's right. Absolutely agree.
Yeah, I'll make that. Okay, so next. Okay, sure. I will get back to you on that.
That's actually really, really important, yeah. Because there's a lot of content that could be indexed, that should be indexed.
So the new features that that we are going to add to the LDP is going to be dataset submission tool, which is a new component for LDP. We are going to add substructure search for small molecules and dynamic linking of apps between small molecules, data sets, and stuff like that. And then there's going to be a list feature, list view. So list feature, which means you can select a list of small molecules and see the data associated with it. So these are all the things that we are working on. And we are also working on making a summary pages based on the category, data summary pages based on the category, like proteomics landing page, genomics landing, transcriptomics landing page, stuff like that. And we have improved our APIs towards the Smart API. So we are working with Smart API people to make sure our APIs are compatible with them, so that it can be reused. And the next thing that would come, okay, yeah, next thing that would come from us is going to be data submission tool, as we discussed. And the main features is going to be, you could submit dataset metadata, assay metadata, reagent metadata, and you could upload a data file, and you could even submit experimental metadata, experimental details about the data set, And we are using Cedar for most of the back-end work in designing the data submission tool. We have already, so the current status, we have already created single sign-on authentication between Cedar and us, so you don't need to register 2 times. And there's an authentication that everyone has to register. I mean, for links, user has to register to submit any data set. And we presented the prototype in Cedar EAB, and this is the one that's going to come up next. So that's all I have.
Right, thanks, Amar. Very good. So, do you guys have any other questions about this? I mean, it's quite a bit of content and detail, and of course, if you do not have any questions right now, you can of course always email any of us, and we are also happy to walk you through any of that, if you will. So Amar, do you want, should we share the dev version for folks to, I guess whenever it's ready, we're going to share it. I think it's ready now, but I'm not sure if he wants to maybe add a few more little things. Then we could share the dev version if you want to, and give everybody like basically an opportunity to provide feedback, if you want to.
And one of them actually meeting said the attack seek and some of these essays are missing here on the landing page, right? So I think that's so whenever you notice a bug like that, can you please provide us the feedback using this button here? There's a provide feedback button. You just click. You can say something like, "Okay, I'm missing this," and sometimes we don't really notice those because of various reasons, right? So if whenever you have time and you notice an issue, please provide us the feedback. And you can track those issues as well. We have a GitHub with issues, tracking those issues as well. So in case you want to follow that, you could do that too.
So we do have a fairly good process, that development and different versions for functionality and content. And then when we put something in release, but of course it's always possible that there is some bug, right? Almost all software doesn't come out bug-free. Yeah. So it's very important that we get that feedback. And in the development version, that would be great. So we can share that with you probably either today or early next week.
Yeah, early next week is a good idea.
Okay, let's do that then.
Okay.
All right, so no other questions. Then thanks everybody. Thanks again, Ned, Caroline, and Amar. And I think there will be a meeting then at the end of March. All right. Thanks, everybody.
Thank you.
Thanks, everyone.
Thank you.
Bye.
Thank you.
Bye.